Like the topic I will be covering today for this webinar is comprehensive DIA workflow in PEAKS 11. So first we'll begin with the webinar agenda. First, we'll be looking at the background of DIA. Afterwards, I'll discuss DIA workflow in PEAKS 11. Next, I'll cover DIA workflow application using 2 cases. And then I will follow up with a summary of PEAKS 11 DIA workflow. And following the summary, I will do the software demonstration. Before I discuss the background of DIA, I will describe the process of DDA, data-dependent acquisition, something we're more familiar with. In DDA, as we know, the mass spectrometer selects a fixed number of the most intense precursor ions in the survey scan and fragment each precursor ion within a narrow isolation window for MS2 analysis. And because of this, typically identification and quantification are biased towards abundant peptides. While in DIA, data-independent acquisition, between every 2 survey scans, the mass spectrometer cycles through a set of predefined precursor ion isolation windows to cover the mass range of interest. In each isolation window, typically around 10 dalton, all the precursor ions are fragmented, and MS2 spectra containing the fragment ions from all the precursor ions in that isolation window are acquired. And this process continues until all the isolation windows are completed. Because MS2 spectra in DIA are consistently acquired across all the isolation windows, a complete record of all peptides in a sample is produced, and compared with DDA, DIA data are more reproducible. Following this process, as we can imagine, MS2 spectra in a DIA dataset are typically complex from the multiple co-eluting precursor ions in the wide isolation window. Unlike DDA, there's no direct relationship between a precursor ion and its fragment ions, since the fragment ions in DIA MS2 spectra come from multiple precursor ions. There are 2 main strategies in identifying the peptides in DIA MS2 spectra, spectrum-centric and peptide-centric. Spectral-centric strategy is typically used in DDA database search to identify the peptide in each MS2 spectrum. To apply the spectrum-centric strategy in DIA data, pseudo MS2 spectra have to be generated first for each precursor ion in the DIA data by assembling precursor fragment groups based on the elution profiles of the precursor ion and fragment ions. And then a conventional database search is followed to identify the peptide in each pseudo MS2 spectrum. On the other hand, the peptide-centric strategy seeks to find the evidence of peptides in the MS2 spectra. In the peptide-centric strategy, the M/Z and retention time of the peptide, as well as the M/Z and relative intensity of fragment ions in the previously generated library, are used to extract the peptide signals in the DIA MS2 spectra. The generated library could be either spectral library or in silico library created from a protein database. In PEAKS, both strategies are using our computational approaches to analyze DIA data. One computational approach to identify the peptide in PEAKS 11 is to use the Spectral Library Search. Spectral Library Search a spectral library is typically acquired from multiple fractionated DDA analysis of the same sample or same type of sample that you use for your DIA analysis on the same instrument, and search against a protein sequence database for identification. In PEAKS, PEAKS first performs feature detection on raw DIA data to find peptide features and associate their corresponding MS2 spectra based on the co-eluting profiles of the fragment ions and precursor ions. And next, MS2 spectra associated with a peptide feature are identified by matching the fragment ion peaks in MS2 to the peptide fragment ion pattern in the spectral library. By applying both strategies, we were able to cross-validate the identification. One advantage of the spectral library search is it requires limited search space, compared with another computational approach in PEAKS, which is shown in the next slide. Using the spectral library search, an argument can be made that there could be bias towards peptides that have already been identified in DDA. Furthermore, there may be people who prefer not to spend resources or time in creating the spectral library from DDA runs. In PEAKS, another approach we offer to overcome those issues is the direct database search approach. In the direct database search approach, instead of using a library generated from result in PEAKS, instead of using in the direct database search approach, instead of using a library generated from experiments, an in silico library is created from a protein database through deep learning. A protein sequence is first clipped into peptides according to the enzyme cleavage rules specified by users, and then PEAKS applies an internal spectrum prediction model and index retention time prediction model on those peptides, and builds a projected spectral library. And finally, PEAKS conducts spectral library search on the generated in silico library. And again, both peptide-centric and spectrum-centric strategies were applied to cross-validate identification. Compared with the spectral library search approach, direct database search offers the possibility of identifying peptides that are not included in the spectral library, but it does requires a significant amount of time to predict the theoretical spectrum. To combine the advantages of both computational approaches in PEAKS 11, we proposed streamlined DIA workflow for peptide identification, integrating those two approaches. This streamlined workflow was first published in the paper shown below last year. It integrates spectral library search, subsequent database search, and de novo sequencing to increase the sensitivity and accuracy in peptide identification, while also limiting the search space, while also limiting the search time. Here, I want to explain our streamlined workflow, giving more details. First, DIA data are searched against a spectral library, which is generated using DDA data. And after false discovery rate estimation, FDR estimation, peptides that pass certain FDR threshold are saved in the spectral library search result. MS2 spectra that couldn't be matched to any peptide in the spectral library within the FDR threshold are brought forward for database search. Same here, peptide that pass certain FDR threshold are saved in the database search result. And any unmatched spectra from the spectral library search and database search are analyzed using de novo sequencing. And in the end, a list of peptides identified by the spectral library search and database search are used to build a new library. And the final search of the whole dataset is performed against this new library. By doing this, a unified global FDR is used to reconfirm the identified peptides to ensure peptide identification accuracy. This is how peptides are identified in the streamlined workflow using PEAKS 11. These three approaches can be applied together in a workflow or separately with the spectral library search or the database or the direct database search or just de novo sequencing. But our proposed streamlined workflow, which performs the spectral library search, the database search, and de novo sequencing consecutively on a DIA dataset, does provide the highest possible sensitivity, which I will show later in the case. In addition, the spectral library search result and database search result can be carried forward for label-free quantification. In PEAKS, unlike the area of extracted ion chromatogram as MS1, which is used for quantification in DDA workflow, the area of extracted ion chromatogram at MS2 level is used for quantification in DIA workflow. To ensure the peptide abundance calculation accuracy, at least 3 high-quality fragment ions that follow the same elution profiles as the precursor ion are used to calculate the peptide abundance. The data in the first case are from a benchmarking paper published in 2022. In this study, different DIA data analysis workflows performed by 3 different software tools were compared by analyzing DIA data of tryptic peptides from human embryonic kidney 293 cells. 3 replicates were prepared at each sample load and measured on orbitransfusion. We used the data from the sample load of 100 nanogram to show the performance of our streamlined workflow as well as other available workflows in PEAKS 11. Before I show the results, I want to take this project as an example to show how the DIA data were analyzed using the streamlined workflow I mentioned earlier. The sample here is HEK 293 T cells. For the construction of the spectral library, peptides were fractionated and 10 DDA data files were collected and measured on orbitransfusion. To generate the spectral library, these 10 DDA data files were searched against a uniprot human database in PEAKS 11. The parameters used to set up the project are listed at the bottom here. Meanwhile, 3 technical replicates were prepared and acquired in DIA mode on the same instrument. DIA data were searched against the spectral library created earlier and followed by subsequent database search using the same parameters used to generate a spectral library. Any unmatched spectra from the spectral library search or the database search were analyzed using de novo sequencing in the end. First, let's take a look at identified proteins. The table here is from the summary page, which you can find in any project in PEAKS. It lists information like the identified peptides and proteins in each of your sample in the project, as well as the total numbers in the project. The total identified protein group number in PEAKS 11 using the streamlined workflow is around 4,700, very close to the number reported in the paper in which DIANN was used. The Venn diagram here shows the common proteins and unique ones identified in each sample. And we can see over 95% of the proteins were identified in all three replicates, showing the superior identification reproducibility of data acquired in DIA mode. Next, I will use this dataset as an example to compare our streamlined DIA workflow with the Spectral Library Search or the direct database search in terms of identified peptide and protein numbers to show why we have proposed the streamlined DIA workflow. The results are shown in the bar graph here on the right. So clearly, the Spectral Library Search followed by the database search increased the peptide identification. When compared to the identified peptide number in the paper, around 18,000 peptides were identified using DIANN. The identified peptide number using the streamlined workflow in PEAKS 11 is significantly higher. We can see over 60,000 peptides were identified in PEAKS 11 using our streamlined workflow. Similarly, the Spectral Library Search followed by the database search increased the protein identification, and compare with the identified protein group number reported in the paper, the identified protein group number using the streamlined workflow in PEAKS 11 is really close. One possible reason why PEAKS identify a significant higher number of peptides but similar number of protein groups could be from the algorithm PEAKS uses to map the peptide to the potential proteins. When taking a close look at the common and unique identifying proteins or peptides in each workflow, we can see an additional 277 proteins were identified in the database search result following the Spectral Library Search. At the peptide level, the database search after the Spectral Library Search increased peptide identification by around 10%. And additionally, around 1,200 peptides were identified in the de novo sequencing step when setting the ALC average local confidence threshold to 85%. And this table here shows the result of de novo sequencing in the last step to analyze any unmatched spectra from the Spectral Library Search and database search in PEAKS 11. So users have the option to export this de novo peptides detected in DIA data to construct the spectral library, as you can see here. But we do suggest users set the ALC between 70 to 85 to use high quality identified de novo peptides. The data in the second case are from a benchmarking paper, which listed on the top right. So in this study, different DIA acquisition methods were compared using a complex proteomic standards. 8 different concentrations of universal protein standards were spiked in E. coli digest, and 3 replicates were prepared at each concentration and measured using 4 DIA acquisition methods on orbitrap fusion. To keep things simple, I use the data acquired in the narrow window to show in a complex matrix. In this case, the matrix is E. coli. How well the UPS1 protein standard was quantified at concentrations from low to high in PEAKS11. UPS1 is composed of 48 human proteins. I will mainly look at the quantification numbers and quantification reproducibility at each concentration, as well as the quantification accuracy using the pairwise comparisons. It's a simple case, but it can address the most relevant information with regard to quantification. First, let's take a look at the quantification number at the peptide level. For one peptide to be quantifiable, an intensity from that peptide had to be measured in all 3 replicates. The top left figure here shows the number of UPS1 peptides quantified at each concentration based on the 3 replicates. And then the coefficient of variation, the CV, based on the 3 replicates, was calculated for each quantified UPS1 peptide at each concentration. And the median CV across all quantified UPS1 peptide at each concentration is plotted in the bottom left figure. As expected, a higher number of UPS1 peptides were quantified as the concentration increases. On the other hand, the median CV dropped when the concentration increased, meaning the quantification is more reproducible at higher concentrations, which is not surprising since it's possible the signals and noise are hard to distinguish at lower concentrations. The figure on the right side is part of the result in the paper. We can see the quantified peptide numbers in PEAKS11 is close or higher than the numbers reported from DIA or Spectral, depending on the concentration of UPS1 proteins. The same criteria apply at the protein level. For one protein to be quantifiable, an intensity had to be measured in all 3 replicates. Similarly, a higher number of UPS1 proteins were quantified as the UPS1 concentration increased. On the other hand, the median CV dropped when the concentration increased. And when comparing the quantified protein numbers in PEAKS11 to the numbers reported in the paper, the quantified protein numbers in PEAKS11 is close to what were reported from DIANN in the paper. When looking at the result from pairwise comparisons of 2 concentrations, 3 replicates in each concentration, the left volcano plot shows the result comparing the UPS1 proteins at 50 fetomole and 10 fetomole, while the right volcano plot shows the result comparing the UPS1 proteins at 5 fetomole and 1 fetomole. The expected ratio in both cases is 5, but we can see the measured ratio of UPS1 proteins were more scattered for the comparison of lower concentrations differing by 5-fold. The mean absolute percentage error MAP to the expected fold change also shows how far the measured ratio is from the expected ratio. The MAP was 8.9% when 50 fetomole and 10 fetomole were compared, but increased to 28.5%, when 5 fetomole and 1 fetomole were compared, showing an overall error increases like when low abundant proteins were compared. Similarly, when applying strict filters when setting up a quantification analysis. In this case, one condition requires 3 confidence samples per group, and another case requires 1 confidence sample per group. The median CV was increased from 7.8% when 3 confidence samples were required per group to 15.5% when only 1 sample was required per group. Though there was an increase in the quantifiable number, I guess here the message here is like we have to consider the trade-off between the accuracy and reproducibility on one hand, and quantifiable numbers on the other hand, when setting up the analysis and interpreting your quantification results. I hope my presentation so far demonstrate PEAKS 11 provides a comprehensive and streamlined DIA workflow using PEAKS 11. Peptide identification and protein identification are increased by performing DIA database search after the spectral library search. And in PEAKS 11, de novo peptides can be exported for spectral library search. Sorry, de novo peptides can be exported for spectral library generation. Okay, so next I would like to show the software demonstration. Okay, so this is, can everyone see my screen? Just want to make sure.
Yeah, it looks good.
Okay, awesome. So this is how PEAKS 11 Studio looks like, the interface looks like. In PEAKS 11, 2 workflow options are available to analyze DIA data: DIA identification analysis workflow, as well as the quantification analysis workflow, which you will see, example. Sorry. Always make sure. Okay. So as you can see here, after loading your data, first, let's take a look at the identification analysis workflow. As I mentioned earlier, the spectral library search, database search, and de novo sequencing they're all optional steps in the DIA identification workflow. Any combination of these steps can be run, but at least one of these steps must be included in analysis. To include the spectral library search, simply click the button next to library search parameters. And from the drop-down menu here, you can select any spectral library that has been configured in PEAKS Studio. Or you have the option to configure a new library by clicking the new button here. And in the configuration window, click on the browse to select a valid spectral library. And also name your spectral library and library name. And then just simply click add/update button. Similarly, to include database search, click the button next to database search parameters and add the parameters accordingly. So here I'll just select committed methylation as fixed PDM, oxidation as variable PDM. And you will upload your database. And next, same here, click on the button next to the novo search parameters to include the novo sequencing. And after adding like all the parameters, you'll click report. And on this page, like report filters that will be used for to filter the final result can be set. And then you click like finish to, and then PEAKS will start like running your analysis. So it's very easy like to set up the project in PEAKS 11. And the quantification analysis workflow is very similar. So after adding the DIA identification parameters, you will click, so after you add the identification parameters, so you just click quantification to go to the DIA label-free quantification parameter set page as shows here. And the settings are very similar to the LFQ setup followed in DDA database identification. After the analysis is finished, you will see a node created for each step, like in the analysis on the left. As we can see, there are spectral library result, database search result, and the novo sequencing result for this analysis. Before I show the analysis report, I would like to show a few like functions in PEAKS 11 to run additional analyses in the current project. So one option, you probably already see I did that. One option is to create a new analysis by clicking on this new analysis button. Within the data selection window, select the data files that has been already like preloaded in the project, or you can always add new data files. Another option is to modify analysis. To modify analysis, so first click on any analysis node. So for example, the streamlined xenotype swaths analysis. And then you'll see this modify analysis icon. Click that, and you see all the parameters and filters in this analysis. And from here, you can modify certain parameters or apply new filters in analysis without having to go through each of those settings again. Next, I will show the result in the DIA identification workflow. To view the analysis report, just double click any node on the left side. For example, the Spectral Library, which is already open here. The report contains a summary page a protein page, and a peptide page. The summary page is similar to DIA DDA database identification page, summary page. At the top of the summary page, protein filters can be set. Under protein page, a list of proteins is shown here. Under the peptide page, it shows the peptides that have the filters used before. For each peptide, for example, let's select the first one. So at the bottom, so PEAKS offer a mirror plot view to show the alignment of fragment ions in DIA MS2 spectra to the fragment ions, to the peptide fragment ions, in this case, in the spectral library. And at the bottom, you can see the ion match table listing the M/Z of fragment ions in the library spectrum, as well as the matched fragment ions within the mass tolerance in the query spectrum. And on the right side, you can easily view the extracted ion chromatograph of the precursor, as well as the ion extracted chromatograph of each identified fragment ion. So they are available for users for further assessment.